Now I have the pleasure of introducing Henry Fabian Thomas. Henry obtained his master's in biochemistry with Martin Lieb in Vienna and is now a PhD student in the laboratory of Krista Buecker at the Max Perutz Labs. Henry's talent as a student has provided him with funding to support his research from several different sources, including an Erasmus Plus grant during his master's and now during his PhD and UniDocs Fellowship, which aims at financing excellent doctoral candidates. I'm excited to hear from your work today, Henry. So please go ahead.
All right, thanks a lot. So welcome to today's seminar also from my side here in Vienna. And thanks a lot to the organizers for putting together this series. I think it's really a great platform to talk science and exchange ideas in these quite challenging times. So as you already heard, my presentation is lagging. Just give me a second. Oh, here we go. Okay, yeah, so I'm a PhD student in the lab of Krista Buecker, and in the Buecker lab, we're fascinated with the process of development, how a single cell can give rise to an entire organism. Of course, during this process, gene regulation needs to be tightly regulated. And as you all know, the very first step of gene expression is transcription, where the general transcription machinery assembles at the promoter and then starts producing an mRNA. However, promoters by themselves give only rise to low levels of expression, and a lot of the regulation actually takes place at the levels of enhancers. These are DNA sequences that can be distal from the promoter. Upon activation, they're being bound by transcription factors. How exactly they're being bound by transcription factors is something that we will hear a lot more in the upcoming talk. What I want to focus on right now is that they bring along a bunch of co-activators. And these co-activators can then interact with the general transcription machinery at the promoter and mediate efficient expression of the target gene. And upon activation, enhancers also undergo changes in the local chromatin environment, so they become more open, more accessible, and surrounding nucleosomes are characterized by or are modified by characteristic histone modifications, such as the H3K27 acetyl mark. And based on this chromatin profile, we can identify active enhancers in a cell type of interest. However, we do not understand very well how these enhancers activate their target genes mechanistically. There are many open questions, for example, whether all enhancers activate their target via the same mechanism or whether they are different mechanisms. While we do know that many genes are actually regulated by multiple enhancers, we also don't understand very well how these enhancers work together to activate their target. And last but not least, one question that we are especially interested in is how enhancers and genes are being activated during the transition from one cell type to another one. And to address all these questions, especially the last one, we're working with mouse embryonic stem cells and their differentiation into epiblast-like cells. This is an in vitro system, which makes life quite easy for us because we're not limited in terms of cell numbers. It's a quite accessible system where we can easily introduce genetic modifications. And this transition is very rapid, so we can follow it with high temporal resolution. At the same time, it does recapitulate the biologically important transition from the early to the late epiblast upon implantation. So I decided to use the system to focus on a single gene locus, which is the Fgf5 locus. As you see, there are 5 enhancers at this locus, the 4 intergenic enhancers E1 to E4 that are activated upon differentiation, as well as one enhancer within the intron of the Fgf5 gene that we refer to as PE for poised enhancer, because it already shows some signs of activity in undifferentiated cells, but then gets fully activated only upon differentiation. And as you might have guessed from this enhancer landscape, Fgf5 expression is also strongly induced upon differentiation. So now we have a gene that is regulated by multiple enhancers and that is activated upon differentiation. So this is a good model system to answer the questions that we're interested in. But maybe the most important property of Fgf5 is that it is not at all required for embryonic stem cells or for the differentiation. And this is very important to us because it tells us that whatever genetic modifications we introduce at the Fgf5 locus will only affect Fgf5 expression, but will not have an effect on the cell state or on the differentiation of the cells. And this is demonstrated here, where we compare the transcriptome of wild-type cells with an enhancer knockout that has dramatically reduced Fgf5 levels upon differentiation. We see that there are only 4 significantly deregulated genes, with one of them being Fgf5. So this tells us that the enhancer knockout does differentiate normally despite having lower Fgf5 expression, because otherwise we would expect much more drastic changes in the overall transcriptome. Now that we have this system, we want to understand the contribution of each single enhancer element, and to do so, we generate individual deletions. I will start by showing you what happens when we knock out E3 or E4. To begin with, here's one representative example of one of our replicates. There are no major changes in the undifferentiated state or at early time points of differentiation. However, there's a slight reduction at the latest time point of differentiation. We can see the same when averaging over several replicates, and then always looking at the log2-fold change at every time point compared to wild type. So also here, there's only a significant reduction at the very latest time point of differentiation. When we knock out E1 or E2, we again see no changes in undifferentiated states, but Fgf5 expression is not properly induced upon differentiation. And compared to E3 or E4 knockout, we see a much earlier effect already at 24 hours in this particular replicate. And also, if we look at the average of several replicates, we see an earlier effect compared to E3 or E4 knockouts. So taken together, these data show that E1 to E4 are required for induction of Fgf5 expression upon differentiation, and that E1 and E2 are required earlier than E3 and E4. When we now knock out the intronic PE enhancer, we see a very different situation. So the first difference is that PE knockout already causes a reduction in the undifferentiated state, and we see the same shift towards lower expression levels at all later time points. And also the extent of downregulation upon PE deletion is much stronger. So upon PE deletion, we see a roughly 10-fold reduction in Fgf5 expression levels, whereas deletion of the other enhancers only caused a 2- to 3-fold reduction. Since PE deletion had such a strong effect, we then wondered whether PE by itself might be sufficient to activate Fgf5 expression. So we generated a cell line where all the other enhancers are deleted and only PE is left. However, in these cell lines, Fgf5 expression upon differentiation is not properly induced, which you can also see here. So this tells us that PE by itself is not sufficient to activate Fgf5 expression. It does need the other enhancers as well. These cell lines now give us a good tool to understand how PE works together with E1 to E4, because if these enhancers would work together strictly additively, we would expect that the expression levels in wild type would be the sum of the promoter contribution as well as the contribution of the PE and the E1 to E4 enhancers. We can now dissect this, because in the PE deletion mutant, we only see the contribution of the enhancers E1 to E4, whereas in the PE only cell line, we only see the contribution of the PE enhancer. Based on this, we would expect that adding up the expression levels in the delta PE and the PE only cell line should bring us back to wild type levels or to even higher levels depending on how strong the promoter contribution is. However, this is not at all the case. Here at 36 hours, the expression levels add up to 50% at most, and the same is also true for other time points. So this tells us that PE acts together with E1 to E4 in a super-additive way. However, one thing that we were concerned about is that PE might not be an actual enhancer because it resides within the intron, so it might also have an effect on, for example, splicing, mRNA stability, or some post-transcriptional mechanism. To address this, we decided to remove PE from the intron and then reintroduce it upstream of the promoter, where it should only act as an enhancer. And in these cell lines, we can indeed rescue Fgf5 expression. So here in this red, you see the reduction upon a PE deletion that is clearly rescued by these cell lines. So this tells us, or this suggests that PE is indeed an enhancer. And in line with this, when we look at the nascent transcription by doing ProSeq, we also see a strong reduction upon PE deletion in nascent transcription. Again, this tells us that PE indeed regulates transcription, that it is indeed an enhancer. So let me quickly sum up what I've told you so far. So I showed you that Fgf5 is a good model to understand how a gene is activated upon differentiation and how it is controlled by several enhancers. I showed you that E1 to E4 are required for the induction of Fgf5 expression upon differentiation, whereas PE has an effect at all time points. And I also showed you that PE has a very strong effect on the endogenous Fgf5 expression, that it is indeed an enhancer, and that it works together with E1 to E4 in a super-additive way. For the remainder of my presentation, I want to focus on why the deletion of PE causes such a strong reduction in Fgf5 expression levels. The most obvious explanation would be that PE is simply a very strong enhancer. So to test this, we performed luciferase assays with the luciferase gene under the control of the minimal SV40 promoter. We then added different enhancers downstream of the gene and assayed the luciferase activity at 40 hours of differentiation. While our positive control, the OPT6 enhancer, strongly activated luciferase activity, none of the enhancers from the Fgf5 locus caused a very strong increase in luciferase activity. And in fact, we couldn't even detect a significant increase, most probably because these assays are quite noisy. We're currently working on improving them, but what we for sure can take away from these data is that PE by itself is not a stronger enhancer than, for example, E1 or E2. And this is also true if instead of the minimal SV40 promoter, we use the endogenous Fgf5 promoter. So then why does PE deletion cause such a strong reduction in Fgf5 expression levels? An alternative explanation might be that PE regulates the activation of the entire locus so that in the absence of PE, the other enhancers are not properly activated. To test this, we did a chip against the active enhancer mark H3K27 acetyl in wild-type cells and upon PE deletion. So at the PE enhancer itself, the K27 acetyl is strongly reduced. This is what we would have expected, so this serves as a nice control. However, both E1 and E2 show wild-type levels of K27 acetyl at the enhancers. So this tells us that these enhancers are properly activated even in the absence of PE, so PE does not seem to control the activation status of the other enhancers. So also these results cannot explain why PE deletion causes such a strong reduction. But to solve this question, I want to take you back to the PRO-seq data that I showed you before. Because if we look closely, we see that there's a lot of nascent transcription ongoing at the PE enhancer, much more than what we see at the enhancers E1 to E4. This is also quantified here. One confounding factor might be that PE resides within the intron, so that every polymerase transcribing the gene must pass through PE. Therefore, we also wanted to do this experiment in the cell lines where PE is upstream of the promoter, so in the knock-in cell lines that I showed you earlier. And there we also see that the PRO-seq signal at PE is stronger than at the other enhancers. We were then wondering where this polymerase comes from, and we hypothesized that there might be an initiation event ongoing at the PE enhancer. And to test this, we did PRO-cap-seq to look at initiating polymerase. And indeed, we saw a very strong initiation event at the PE enhancer, once more stronger than what we see at E1 to E4. But at the same time, we do not see any stable transcript arising from PE. So PE is indeed an enhancer and not, for example, an alternative transcription start site. So taken together, these data show that there's an accumulation of initiating polymerase at the PE enhancer. And we think that this polymerase might contribute to the expression of Fgf5, and that this is why upon deletion of PE, we see such a strong decrease in Fgf5 expression. By that, I want to conclude what I told you today. So I showed you in the first part of my talk that at the endogenous locus, PE strongly activates Fgf5 expression. However, PE by itself is not sufficient, and it does need the presence of the other enhancers, E1 to E4. I also showed you that in luciferase assays, PE by itself is not a particularly strong enhancer, and that it doesn't influence the activation status of E1 or E2. And I showed you that PE accumulates high levels of initiating RNA polymerase, and that we think that this contributes to Fgf5 expression. Of course, there remain many open questions. One of them is why PE accumulates such high levels of RNA polymerase. This might be a sequence intrinsic feature, so PE might be bound by certain transcription factors that just bring along a lot of polymerase. However, very preliminary results that we have suggest that the distance to the promoter might also play a role, so that having an enhancer with open chromatin close to the promoter with its high level of RNA polymerase might be sufficient for an accumulation of RNA polymerase. The second question is how this accumulation of POL2 would contribute to Fgf5 expression. Once again, there are several possibilities. One is that PE simply acts as a reservoir for RNA polymerase so that it recruits the local concentration of polymerase that can then be recruited to the promoter by the outside enhancers. An alternative explanation could be that the C-terminal domain of the RNA polymerase that has been reported before to be involved in interactions with transcription factors and other polymerase molecules, so that this CTD might contribute to the recruitment of additional transcription factors, cofactors, and polymerases, and that this then leads to Fgf5 activation. By that, I want to conclude my talk. I want to thank my entire lab. Here you can see us on a very normal workday. We have a very strict dress code here in Vienna. I especially want to thank Christa, who's a great mentor, as well as Swati and the former students Elena and Axel, who contributed a lot to this project. I also want to thank Ursula, who helped me with setting up the Pro and Pro Cap-seq, and Joanna and her lab for getting this project started. And of course, I also want to thank you for your attention. And before answering your questions, I just want to mention that we recently posted the story on bioRxiv, so if you want to read it and get back to us with some feedback, we'd be very happy about that.
All right, excellent. Thank you very much, Henry. That was a fantastic talk. Just for anybody joining us for the first time, I just want to quickly remind you that if you have questions, we have two ways of doing things. You can either write your questions on the Q&A box, or you can raise your hand and we can unmute you so you can ask your question live to Henry. So in the meantime, while people start writing their questions, can I just ask you real quick? So when you mentioned that you have some preliminary evidence for the POL2 abundance in the enhancer region, do you mean like, have you done any chip-seq for POL2? Do you see an accumulation also, or some kind of turnover happening at the enhancer?
Well, we didn't really have any data on the turnover yet, but what we do see is that so these are all the enhancers, and we see that enhancers that are closer to the transcription start site seem to have a higher pro-cap signal, so they seem to have a higher initiation of RNA polymerase. And if we look at P300 just as a control for the enhancer strength, this does not show the same trend. So that's why we think that being close to the promoter might contribute to accumulating a lot of polymerase.
All right. Okay. So questions start coming in first. Would you mind going back to your QR code? So without the pointer over it, I think some people are interested in scanning it. So if you can just maybe leave it in there while I continue asking the questions. All right. So we got somebody with their hand raised. Ignacio, if you want, go ahead and ask your question.
Okay, great. You hear me, right?
Yeah, I can hear you.
Great. So thank you, Henry, for the talk. A quick question is, I was wondering about the actual transcription factors regulating this gene Fgf5, and if the motifs in those enhancers and the motifs logic in those enhancers is common. I wonder whether you're deleting the full enhancer or specific motifs in each of those enhancers.
We're usually deleting the entire enhancer, so based on the P300 and the HVK27 isotope peaks. We also want to look at the transcription factors at some point, but so far we didn't really look at any specific motifs.
Okay, great. Thank you.
All right, thank you, Ignacio. We got a couple of questions on the Q&A. So Ben Weekly is asking, what are the key transcription factors specific to this differentiation? Could you do a motif analysis to see if they will be expected to bind to PE?
Yeah, so I mean, we didn't really go into the transcription factors yet. The problem is that we're just really looking at very few enhancers, so that makes it quite tough to do a proper motif analysis. It is something that we want to look into, but so far we didn't really analyze which transcription factors bind there yet in much detail. I mean, we know that there's some OCT4 and OTX2 binding, but besides that, we didn't really go much in detail.
All right, okay, thank you. So Pallavi Mustafi wants to know, how far do you think you can relocate the PE from its endogenous location where you would still see the effect of transcription?
Yeah, we're currently trying to generate cell lines where we do move it further away. I don't really have a very good idea of what would be the critical distance, but we would assume that moving it further and further away would reduce Fgf5 expression. But yeah, I mean, I don't really know, also because at some point we have to take into consideration the 3D organization of the genome. So we don't really know like which actual distance the genomic distance would then correspond to. But yeah, we're in the process of testing this, hopefully.
Okay. Vicky Metsis is asking, very nice talk. Thank you, Henry. How different are the sequences at each enhancer? Do you think their differences are driven by sequence, by position, or both?
Well, I mean, I think the position does have an effect. At the moment, we can't really exclude that the transcription factor, like the motifs themselves, also have an effect. So we don't really know yet, but I would think that the distance stuff does have a very strong effect.
Okay, so Ioannis Tsagakis is asking, thank you very much for a clearly explained talk. Would probe capsid be capable of detecting a short-lived enhancer RNA? And there's a second question that says, perhaps I missed it, but why does removing PE from a downstream position to an upstream position not have a different effect?
Well, so I mean, with PROSEC, we would also detect enhancer RNAs because we look at the nascent transcription. So we would basically detect any ongoing transcription, no matter how unstable the product is. I'm not quite sure whether I got the second question.
He's not quite sure why moving the PE from the downstream position to the upstream position doesn't have a different effect, but I don't know where you want to go.
I mean, our reasoning was that if we move PE up, like away from the intron, then we would get rid of any effect that PE might have on, for example, splicing or RNA stability. But in theory, it's like just for the enhancer itself, it shouldn't make such a big difference whether it's positioned upstream or downstream.
Right. Okay, so so there's a couple more questions, but I'm going to ask you if you can take a look at them later. We're going to go ahead with Srinivas. Thank you very much, Henry, again for your talk.